Hello, my friends, and welcome to a video on cell-mediated immunity. We did the overview on adaptive immunity, and we're going to break it into cell-mediated and antibody-mediated. This will be cell-mediated. We've already talked about how incredibly intertwined the two are, so don't think they're all that separate. They're really two pieces, but we will tease out this video to explain cell-mediated, but the last one ran super long. I apologize, but it was packed full of core information that's going to help you to understand this much shorter video. To test ourselves, we'll take these items here, and this is a review of last module, except we're only pointing out the ones that are cell-mediated. We'll just point out the ones that are cell-mediated here, just as purely review. And then what may be, possibly, arguably, the most difficult test yourself that I think I've ever made is to order these. And this is the whole order from A to Z, if you will, from 1 to 11. These are out of order. We'll put them in the correct order. This is how the entire cell-mediated immunity works. And the good news is 1 through 5, when we put 1 through 5 in the correct order, are going to be identical to the next module on antibody-mediated immunity, because the first 5 steps are the same, all about activating helper T cells. And then from 6 to 11, it splits depending on whether you're talking about cell-mediated or antibody. And it's really tempting to write this module in a chronological order. First this, then that. But it's just, immunity is just so complex and interwoven that I'm resisting teaching it in a 1 through 11, but I will test ourselves. We can test ourselves afterwards in this sort of 1 through 11 way, or ordering the steps of cell-mediated immunity. Remember, adaptive was humoral or antibody, and cell-mediated, we're going to get rid of antibody for now and deal with it in the next module. Now we'll talk about cell-mediated, which is going to deal with endogenous antigens, foreign antigens that have gone inside of your body cells and infected your body cells. And that means that the cytotoxic T cells have to become activated, and they have to become immunocompetent, and they've got to go and they've got to destroy your infected body cells. So on this side of cell-mediated immunity, we're really talking for the most part about your own body cells that have been infected with a virus. COVID-19, CoV-2 virus, been infected with a bacteria, E. coli, Staphylococcus aureus, name them all, Heliobacter, whatever it is you want to deal with. Or could be cancerous cells. The cytotoxic T cells and the cell-mediated immunity are really good at taking care of cancerous cells. And the idea with both cancerous cells and with infected body cells is to keep them from proliferating, stop them in their tracks, and keep them from spreading. And that's what we're all about. So to start off, you have to activate both the helper T cells, and that's also the exact same in antibody-mediated immunity. You have to activate the helper T and the cytotoxic T. And remember, helper T cells are often referred to as CD4s, because that's the key protein on helper T cells. Cytotoxic T are referred to sometimes as CD8, because that's the key protein on cytotoxic. There's a 2-step process to activate both. And in 1 and 2, this is your inactive helper T cell. It all starts with a helper T cell first. You're going to do helper T first and then cytotoxic T. When we go to antibody immunity, what are we going to do? Helper T first and then B cells, but it always starts with helper T. Here's an inactive helper T, just waiting to be activated. It's sitting in the secondary lymph organ, the spleen, or in the lymph node, and it's waiting for an APC, antigen presenting cell. Remember, that APC binds an endogenous or binds an antigen, presents it on the cell. The antigen is shown as this diamond, the diamond, and presents it to the helper T cell, which is inactive. But at the presentation, remember the presentation, and it's going to show you here, between the MHC molecule, I'll show an arrow, of the APC, and the antigen, it's the CD4 of the inactive T cell binding with it's this binding right here, the CD4 with the MHC. And that binding is constant. That's like a setup. That's like making a date with somebody. Are you going to like that person? Are you going to hit it off with that person? Are you going to interact with that person? Are you going to date more? Are you going to move your relationship forward? Who knows? But setting up the date is important, because there'll be no chance to do any of that if you don't set up the date. And that's a way maybe you can think of the CD4 and the MHC2 coming together. They're setting the table. Tortori uses a start the car for MHC2 and CD4. Turn the key in the car. that does not confer driving. In order to drive, you need to shift it into a gear, and that's the TCR antigen. That's the second part. So I'm using a dating analogy. To set the table, you have to have that date and you have to meet that person. Where it goes from there, are you going to have the right TCR bind to the right antigen? Who knows? But you have to set the table with the CD4 MHC2 binding. And if the TCR, the T cell receptor on the inactive T, does match that antigen, if it's that 1 in a billion match, then you know that that is the correct helper T cell to clonally select and blow up into billions and trillions of copies. Second step is really straightforward. It's called co-stimulation. Some factors, mostly interleukins. I am not going to hit you with this hard. Mostly interleukins are released in a way to activate the inactive helper T cell. And so antigen binding to the TCR plus co-stimulation equals activation. And just pay attention again. Pay attention to that MHC2 on the APC and the CD4 on the inactive helper T cell, and those two coming together. Again, regardless of whether the antigen and the TCR are a good fit, you're going to have a date. You're going to at least get together and say, hey, is this a match? The presentation. Hey, is this what you're looking for? Yes or no? Yes, great. No, hey, no big deal. We tried, right? Rather than the cells randomly going all over the place looking for each other. That APC is going to present the antigen and say to the inactive helper T cell, hey, is this what you were looking for? And that's that whole lock, key, and a lock. And if it is a match between the antigen and the TCR, then boom, you know you have the right helper T cell. Throw in a little co-stimulation, and ba-bam, you know it's time to clonally select and blow those few hundred copies or 1,000 copies of that inactive helper T cell that has the perfect TCR to bind the antigen, and ba-boom, from 1,000 to 1,000,000,000 as fast as possible, because the clock is ticking and you may be dying. So let's get them cranked up, right? And that's what that clonal selection is. That's part one. And again, easy thing for you is that the next video will start exactly the same as this video. You'll do exactly the same thing to start off and activate the helper T cells in order to move to antibody and B cells. But here we'll stick with cytotoxic T cells. Okay? And what you're going to do is you're going to use a massive amount of clonally selected helper T cells, the correct helper T cells, to then clonally select massive amounts of inactive cytotoxic T cells and get them fired up too. So here's the infected body cell of which we were talking. There's a virus, a CoV-2 virus inside, right? There's some sort of a norovirus inside this cell. It's destroying the cell from inside. There's a Heliobacter bacteria inside of it. It's destroying the cell from inside. This cell is going to, the antigen's already endogenous. Go back to the previous module to look at the details of processing and presenting an endogenous antigen, breaking it apart, putting the MHC1, now it's MHC1. I'll talk about why it's 1 and not 2 in just a second, and presenting the antigen. This time the antigen is shown as a circle. Now the MHC1 binds to the CD8, and that's again a match, like setting up a date. And lo and behold, look, on the inactive cytotoxic, you have a perfect TCR, that 1 in a billion TCR to match this antigen. And a little more co-stimulation this time, they are going to say, by interleukin 2. That's what IL-2 is. Don't sweat the co-stimulation. Understand it's the important second step, but don't get too hung up on it, right? The antigen binding is the first step. The co-stimulation is the second step. If those two work out, you're going to take these few thousand inactive cytotoxic T cells that happen to have the exact correct TCR for the antigen, and you're going to just blow them up into trillions. Okay? And remember the two lines, the effector line and the memory line. When we get to the next module, we'll be looking at this with antibodies and B cells. But for now, let's stick with cytotoxic T. So again, you've got helper T cells, co-stimulation, binding, clonal selection of the exact cytotoxic T cells that you need with that perfect TCR or that T cell receptor. And that cytotoxic T cell is going to be clonally selected, and billions and trillions of copies. They will head out of the secondary. Remember, the clonal selection will happen in the secondary lymphatic organ, in the spleen or in the lymph nodes. Once the clonal selection happens, those cytotoxic T cells are arced up and ready to go, and they're activated. They're going to head out to the infected tissue like a beeline, and they're going to be looking for cells to present them. They can looking for cells that present the antigen, looking for antigen presenting cells, which are infected body cells. They're going to take off into the tissue and find as many infected body cells as they can, and they're going to destroy those infected body cells. So I will slow down just for a second to back up, because when we did the first step, inactive helper T cells binding to an antigen presenting molecule and then making that match, it was MHC2 and CD4. Right? MHC2 is specifically, you have that antigen presenting cell. It's presenting a foreign antigen to the helper T cells, showing it a foreign antigen. And MHC2 and CD4 are associated with foreign antigen presentation. Now, that doesn't happen when you activate the cytotoxic T cells. The cytotoxic T cells are going to be activated by an infected cell of your own, not a bacteria, not a virus, an infected cell. So the bacteria or virus that was originally, where the antigen of a bacteria or a virus was originally dealt with as an exogenous antigen by an APC and presented to a helper T cell. Now you've got that antigen inside of one of your cells, your own cells. It's a self cell. It's one of your lung cells. We'll talk about COVID in 2 modules about the type 2 septal cells. That's where COVID goes. The type 2 septal cells in your alveoli. Those are your cells. So this is an infected body cell. It's one of your own. So the antigen is going to be some sort of a mutated self-antigen. But this connection here, I want you just to understand, it's going to be MHC1 and CD8. Instead of MHC2 and CD4, it's going to be MHC1 and CD8. And these two are more associated with your own cells, with self-antigens. Right? But the antigen is kind of warped and sort of weird, and it doesn't really fit because it's some sort of foreign antigen presented on an infected body cell, or it's a mutated cancer cell, and so you do need to make that match right here. And if you're confused as an MHC2 with CD4 or CD8, I just remember multiply them together, and if you get 8, you get the right answer. So MHC2s go with CD4. 2 times 4 is 8. Sorry. MHC1s go with CD8. 1 times 8 is 8. That's all I got. But anyway, just be aware of that it's a slightly different, but it's the same thing. It's making a date. It's the CD8 and the MHC1 getting together and saying, hey, is this what you're looking for? Is this infected body cell antigen what you're looking for? And that allows the cytotoxic T cell, the one with the correct receptor, to be the right one and to be clonally selected into effectors, which are the cells that are going to go out and do all the damage, and memory that are going to hang on. And we'll talk a little bit about memory in the next module, what the memory cells do. Okay? And then all that remains, no big deal, is to actually kill the infected body cells. No big deal. We got this. And so here's your activated cytotoxic T cell, all ready to go, takes off into the tissue, barrels into the tissue, finds an infected body cell. Why is this match? Well, because this is the perfect TCR for this antigen. And that's already been confirmed by the binding with the MHC and the CD8. And again, here's the MHC1. And here's the CD8, and they're locked up together. Oh, this happens to be the correct antigen. No surprise, this isn't chance. We've built billions and trillions of copies of this particular cytotoxic T because it is the correct TCR to bind the antigen. And then once the binding happens, granzymes are released. What do granzymes do? They go into the infected body cell, your own cell, and they stimulate or they induce apoptosis. They get that cell to blow up. Right? And that releases all the microbes back into the tissue, into the interstitial fluid. Now they're fair game. They were protected inside your body cells. That's what microbes do. That's what bacteria and particularly viruses like to do. They hide in your cells. Well, now the apoptosis is going to cause your infected body cell to blow up, release the microbes free into the tissue, and now standard phagocytosis can go to work. Your neutrophils and your macrophages can just non-specifically gobble up all these freed microbes. They would have been much more dangerous inside of your infected body cell. Or on the other hand, you can kill the infected body cell a separate way. You can release perforin, and that will go in, or perforin, either way. You can say it either way. Perforin or perforin. I'll go with perforin. You can release perforin into the cell, and perforin will act like a MAC. Do you remember what an MAC was? A membrane attack complex. It was one of the ways that complement proteins killed cells. The perforin acts very similar. It just injects a tube or perforates. It pokes holes in the cell, a channel in the cell, and then the fluid will move in and it'll destroy the cell. Okay? And that's called cytolysis. You can go with cytolysis, but if you really want to give it some flair, go with cytolysis, right? And that's destroying these infected body cells. Those are the two ways. Okay? Pretty straightforward. The next module is going to be a lot longer because we're going to throw in a bunch of other stuff about immunological memory, memory cells, and all that, but it's going to follow, the beginning of the next module is going to follow exactly this pattern. So which of these are related to cell-mediated immunity? And again, this is just a repeat of the previous modules. First, test yourself. It is not B cells, but it is T helper cells. That is part of cell-mediated immunity. I'm putting a check next to the ones that are characteristics of cell-mediated immunity, and I'm putting an X next to the ones that are not. Involves clonal selection, you better believe it, into effectors and memory. In this case, both helper T and cytotoxic T. Uses BCRs? Nope. That's coming up next module. Uses TCRs? Yeah. Antibodies? Just wait. It's coming next module. Best on exogenous? Just wait. Next module. Endogenous antigens. An antigen inside of an infected body cell. COVID-2 virus inside of your type 2 septal cells when you have COVID. That's what cytotoxic T cells are really good at. And works best on cancerous, or infected cells. That is also cell-mediated immunity. Okay, here we go. The trickiest, and again, in my opinion, the trickiest of the tricky. So if this seems difficult to you, you're in the right place. Here we go. Let's see if we can iron this out. Remember, the first 5 will deal with nothing other than helper T cells because that's that foundational base. All right. Here we go. All right. Number 10 will be 1. Antigen-presenting cells phagocytose exogenous antigens and express them on the outside of their cell membranes using MHC2. That MHC2 is going to bind to CD4 binding. So as APCs present the antigens to inactive helper T cells using that MHC2 CD4 binding connection. Now we got to get the right TCR antigen. So TCRs of inactive helper cells match that presented antigen on the APCs. That's important. That's kind of step 1. And what was step 2? Well, step 2 was co-stimulation. And I'm not going to do this fancy co-stimulation by IL-2. That's going to be later on. That's going to be the next step. I'll do just co-stimulation. About the most boring thing you can think of. Just co-stimulation. Again, don't sweat it. Just know that there's co-stimulation. That means molecules like interleukin have to be released as a secondary step to get that inactive helper T cell really going. Okay, and now you're going to blow them up. This is clonal selection, okay? Boom. Cytotoxic. No, not cytotoxic. Oh, I almost went for that. Helper T cells. Okay, helper T cells are activated. Boom. Helper T cells, these are the first 5. And they're going to be the first 5 when we do this in the next module too. Helper T cells are activated and undergo clonal selection in the secondary lymphatic organs. Okay, now, let's see. Let's get rid of the ones. This is hard. Let's get rid of the ones I've already used. I remember I do this on the matching too, right? Just to make my life easier because believe me, this is not an easy exercise that I am undertaking and you're going to undertake here too. This is tricky. Why do I not have 5? Here we go. Okay. Okay, now, what comes next? What comes next? Infected body cells present their antigens, the endogenous antigens now, on the cell surface, using that MHC. Typo. Hang on. You didn't see that. You know that 2 and 8 don't multiply together to be 8. MHC1, I beg your pardon, but that's okay. We learn from our mistakes, right? MHC1 to CD8 binding connection, right? That's going to be number 6. What happens now? We need that TCR to match, right? We need that TCR to really kick in, right? Okay, so we need that TCR of inactive cytotoxic T cells to match the presented antigens. That has to happen. And again, then we need co-stimulation. This time I'm going to go with the specific co-stimulation of release of interleukin-2 because it's a little more specific when the cytotoxic T cells are being activated rather than the helper T cells. Remember back with helper T cells, we just said call it co-stimulation and move on. This time we'll say it's interleukin-2. But again, don't stress it. There's like 27 interleukins and you don't need to know all of them unless you're going to be an immunologist. Okay, we're co-stimulated. Now we need to activate and undergo clonal selection. Okay, so this will be number 9. Those cytotoxic T cells that have the exact perfect TCR, T cell receptor, are activated, undergo clonal selection. and then boom, out to the tissue. Effective cytotoxic T cells migrate out to the tissue. They seek out the infected body cells, and they use either granzymes or perforin to kill the cells. And then lastly, number 11 is that don't forget, on the other side of the effectors, the other part of the story is those memory cells, hugely upsized amounts of clonally selected memory helper T and cytotoxic T cells with the exact correct TCRs. They sit patiently in those secondary lymphatic organs and they wait for that secondary infection, or the third infection, or the fourth, or an infection 50 years later, and boom, they're all over it. And that's that adaptive, that learned, and that's what vaccinations are all about. Okay. Thanks so much for sticking with it. On we go to antibody-mediated immunity next. A little trickier, a little bit longer, but we'll work our way through and we'll use a lot of the same principles. So thanks so much for sticking with it. I always appreciate it.